Hello, welcome back to my channel. So last class and the last video, I had started with the fundamental unit of life. Now we'll be continuing with the same chapter. So this is lesson 2 in the same chapter and we'll be learning about the structural organization of a cell. What do we mean by that? At a very basic level, we will learn how cell is organized. So in this lesson, we'll be looking at the cheek cell experiment, another classic experiment. Then we'll learn about the main features in cell and the basic comparison between plant and animal cells we'll do at a very basic level. We'll go more deeper as the lesson progresses, but in this particular lesson, we'll do a basic comparison between plant and animal cells. Then particularly, we will specifically learn about the plasma membrane and the cell wall. So we're going a little deeper into the cell. So first of all, let's look at the cheek cell experiment. Now, this we are doing in order to get a glimpse of what an animal cell looks like, right? So we are human beings, also animals. So we'll be learning, we'll see how animal cells look like through this experiment. Now you'll be doing this probably in your lab also. It's a classic experiment and it's simple to do. So let's see how. In this, basically, you take a scraping of your cheek cells using a toothpick, then spread it on a slide. It's anyway containing moisture, so no need of water, just a drop of a stain. Here methylene blue is used. And with the cover slip, with the help of a needle, the cover slip is used to cover the slide. As we always do, blot out the extra. And pressing with a needle, you have to be careful to be very gentle. Yes. So in the end, you get this picture. So right? This is the animal cell. So we can see again that here nucleus is visible. That is the dense region inside the cells near the center of each cell. So these are clearly different from the plant cells that we observed in the onion peel, right? So we'll see how they are different, but they're also same. How are they same? They have basically 3 main features in both plant, animal and every organism. So what are the 3 main features? First, an outer membrane is present, which is called plasma membrane. Then there is a densely stained area, which is the nucleus. And then in between these, we have cytoplasm. Now this cytoplasm contains a fluid matrix. It's filled with a fluid and also that's called cytoplasmic fluid and it houses other cell organelles. We'll get into that as we progress further in our chapter. But for now, this is the 3 main features that you will see in all cells. Now, in only plant, fungi and bacterial cells, that means not in animal cells, but only in these kind of cells, there is another addition to this. And this is a very major addition because it provides strength to the cells. And that's nothing but cell wall. Right? This is present only in plant, fungi and bacterial cells. Now let us observe these both kind of cells, that is that of a plant cell and an animal cell, and compare how they are different and how they are same. So for that we already have seen a plant cell, right? We have already seen plant cells in onion peel experiment that we did in the last class, and now we have seen animal cells through cheek cell experiment. So I have put these slides together, and how are they same? They are same because in ways like there is nucleus in both the types of cells, and there is also a lightly stained area in all the cells in both these slides, and that is nothing but the cytoplasm. And in the animals, in the cheek cells, if you observe, the boundaries are not so distinct. Whereas in the onion peel cells, you can see that the boundaries are very, very distinct, right? They are so densely stained. That's because of the presence of cell wall in the cells of onion peel. That is, in the plant cells, there are cell walls, right? So the boundaries are very distinct. So these are the ways in which they are different That means they have a cell wall over here. And in case of this particular slide, if you see, the intercellular space is also another point of difference, right? Intercellular means between the cells. Inter always means between. So intercellular space is absent in the peels of onion cells, in the onions peel cells, and intercellular space is present in the cheek cells. They are loosely packed. And one more difference is that onion peel cells are block shaped or rectangular more or less, and cheek cells, they are irregularly shaped, right? So these are the ways in which these both are same and different, but mainly our area of focus is the cell wall in this chapter. So in this lesson, so cell wall is present in onion peel cells or all plant cells, and cell wall is absent in animal cells. Now let's take the first part that we'll be studying in depth in this lesson. So that is nothing but the plasma membrane. So remember where it was? It was the outermost layer, thin layer of every cell, every living cell, okay? So where is it lying basically? It's lying between the cytoplasm and the external environment, right? So it acts as a boundary between the external environment and the cell. It encloses the contents of the cell and separates it from the external environment. So let's look at its definition. It's the thin membrane that forms the outer surface of the cytoplasm and separates the contents of the cell from its external environment, right? And what is it made of? It's made of mainly lipid, that is fat, lipids and proteins. So let's look at the ultrastructure of the plasma membrane. That means a highly magnified picture. So it's like this. So you have two layers of lipid, the fat molecules, and then there are proteins. And these proteins are sometimes attached to carbohydrate chains. So this is the ultrastructure of cell membrane. Now plasma membrane functionally is also called as selectively permeable membrane. Now what does it mean by selectively permeable? What does that mean? It means that the plasma membrane permits the entry and exit of some materials in and out of the cell. It also prevents the entry and exit of some other materials, in and out of the cell. So as we know, the cell needs certain things like gases or nutrients. At the same time, the cell does not need other things like foreign materials which might harm the cell. It permits some good things and it prevents the entry of some harmful things, right? So because it's selecting what should enter the cell and what should not. It's selecting, it's called selectively permeable. Permeable as in it permits or not. Okay, the selective permeable membrane, plasma membrane is also known as. So how does this happen? How do substances move in and out of the cell? To the cell membrane, two ways are there. One is diffusion. In that also subways are there. But then firstly it's diffusion. So simple diffusion happens in case of gases like carbon dioxide, oxygen, nitrogen, and certain fat soluble vitamins like A, D, E, K, right? And when water is involved and when water moves through semi-permeable membranes, we call the process of diffusion in this case as osmosis. Let's look at diffusion in detail. Remember what diffusion is, okay? In basic science, we have, I think we have already learned diffusion. So, the layman's example for diffusion is basically cooking in kitchen. If suppose your mother or you yourself are cooking something very fragrant inside the kitchen, someone sitting at the other room will also slowly receive the smell. So what is happening? The molecules or the particles that are being generated while cooking It's slowly moving into places where it is less. From high to low, it's slowly moving. Do we make it happen? No, it happens on its own. That means spontaneously, right? So that is basically diffusion. It's the spontaneous movement of particles of a substance from a place of higher concentration, that is the kitchen in our example, from a place of higher concentration where it is present more to a place where its concentration is low. So let's look at this example over here, the first one, right? So let's say these are gases. The gas molecules are more. Suppose it's oxygen, okay? Oxygen molecules are more outside the cell and the cell needs it. So what happens? Slowly these molecules move to a place from the higher concentration, that is the extracellular fluid. Extra as in outside. So extracellular fluid into the intracellular, not intracellular. Intra always means inside, to the inside of the cell. It's slowly moving on its own. And this happens until an equilibrium is reached. That means there is an equal number of gas molecules outside and inside the cell. So that's how the substances move spontaneously through diffusion into the cell through the cell membrane, right? So the opposite also happens. That is, if it is in the higher concentration inside the cell and outside it's low, slowly some particles can move on its own to the outside where it is low and till an equilibrium is reached. So this happens in case of gases and fat-soluble vitamins where the particles are small. Now in some other particles, in some other substances, the molecules are very bulky. So in such situations, the proteins, remember in the ultrastructure we noticed proteins on the cell membrane. So those proteins help in diffusion. Sometimes the proteins act as a channel and sometimes as a specific carrier. That means specific proteins are shaped in such a way that it can transfer specific substances through it. So this process by which it's spontaneous, but the proteins help in the diffusion, it's called facilitated diffusion. The first one was passive diffusion. Now both these are spontaneous. It can happen on its own. Now, the second way, as we already saw in the previous slide, is osmosis. And in that, water is involved. So, osmosis is basically the movement of water through a selectively permeable membrane. In this case, biologically in our cells, the selective permeable membrane is nothing but the plasma membrane or the cell membrane. Yeah, so movement of water through a selectively permeable membrane from a region of high water concentration to a region of low water concentration. So remember, here we are looking at the water concentration. More than the amount of water, it's the concentration. When is the water concentration high? When the solute concentration is low and the amount of water is higher compared to the solute. So, in such a situation, water moves through the semi-permeable membrane into a place where lower water concentration is present. That means outside environment until equilibrium is reached. I'll actually show you a simple experiment that I did in my kitchen that you all can do in your kitchens as well. So it's nothing but to observe osmosis in raisins, in dried raisins. Okay. So let's see how that happens. Yes, so day 1, that is night 1 actually, I took some dry raisins and I soaked them in water. Closed it and went. The day gone by, and then the second day I observed what we observe in this, that the dried raisins have swollen. and become larger. What does that imply? It must have absorbed the water from the outside environment, and that's why it became bigger. So just roughly I've shown you how it looks in comparison to another dried raisin. Now I'm putting in some sugar. Just let me confess to you, I didn't just put that much sugar, I added more sugar in that. So if you're trying, make sure you put a lot of sugar. And I put the swollen raisins back into the highly concentrated sugar solution. So what did I observe the next day? Yes, so day 3, the first one that I'm picking up is the one that was in plain water. Now in the middle is the raisin that was put in sugar solution, and at the end, okay, the third one here was the dried raisin. So I did it for you to compare, though it's not the same raisin, but still you can have a rough idea about what has happened. So basically, the dried raisin was small and wrinkled. When I put it in water, the next day it had swollen. It had taken up water from the outside of, outside environment of its own, and then it had swollen up. Then I put it in sugar solution, and again it shrunk, right? In a really concentrated sugar solution when I put it, the next day it shrunk back, right? So now let us see what happened there. Let us just scientifically try to explain. Now I am using like rough diagrams over here. It's not something that, it doesn't look like that in real, but then just for us to understand osmosis. So in the first case, what do you observe? Inside the cell, water concentration is low. Why? Because solute concentration is high. Outside the cell, water concentration is high. Why? Because solute concentration is low. So in a solution where water concentration is higher than that inside a normal cell, such solutions are called hypotonic solution. I'll just repeat it. In a solution where the water concentration is higher compared to the water present in a normal cell, such solutions are called as hypotonic solution. Yes. So what do you think will happen over here? The water will move from the cell and to the cell. It will always keep happening, but more water will move from higher to lower concentration. That is, from outside to inside. So such a type of osmosis where water enters from outside into the cell is called endosmosis. Endo always means inside. So this is called as endosmosis. So gradually what will happen? The cells will increase in size. That's what happened with our raisin. It drank the cells inside the raisin, absorbed all the water, and it got swollen. It became turgid. So that's what happened over here. And then in the second part, you see the amount of solute and water is the same outside and inside the cell. Such solution in which water concentration is the same as that inside the cell. Inside a normal cell, such solution are called as isotonic solution. So in this situation, water same amount will keep going inside and outside, and the equilibrium will be maintained. So basically, the cell will remain the way it is. Okay. Now, another situation is when the solution outside is more concentrated than the solution inside. Right? That means the water concentration is less outside. Such solutions which are more concentrated than the concentration of a normal cell, inside a normal cell, such solution are called as hypertonic solution. Hyper is always more. I know it's a little confusing, but when we focus only on the water while deciding whether it is endosmosis or exo, that is, water if it goes out or inside, that can be decided. If you need to decide that, just focus on the amount of water. So let's focus on that. Obviously, more water will flow from an area where it is more to an area where it is less. What happens is exosmosis. Exo means that already always says outside. So exosmosis happens. That means water flows from more water. Water flows in both directions, but more water will flow outside the cell. So in such a situation, when a cell is put for a long time inside an hypertonic solution, what happens is this. Do you see the cell wall is more or less intact, but the plasma membrane peels away from the cell wall and cytoplasm shrinks because a lot of water has gone out of the cell. This process is called as plasmolysis. So what is it? Plasmolysis is the process by which cells lose water and shrink when put in a hypotonic solution. Why does it shrink? As its protoplasm peels away from the cell wall. That's how it shrinks. So I just read it again for you. Plasmolysis is the process by which cells lose water and shrink in a hypertonic solution as its protoplasm peels away from the cell wall. Just remember these long definitions in bits always. So what happens first? You put it in a hypertonic solution. Then what happens? How does the cell react? They lose water and shrink. So how do they shrink? Protoplasm peels away from the cell wall. So if these 3 points you order it properly, your definition is done. Alright, you don't need to memorize in a rote way. Now let's observe osmosis in a more clear way altogether. These 3 situations altogether we can see. So the middle one is the normal thing, as in isotonic solution where the concentration outside and inside is the same. The cell remains flaccid, that means in a normal way. And when you put it in a hypertonic solution, water flows from where the concentration of water is more inside. Water flows from the cell outside into the external environment and gets plasmolysed. When you put it in a hypotonic solution, wherein the external solution is less concentrated than the internal solution. That means water is more outside, then water flows into the cell where it is less and it becomes turgid. Now, turgid is another word for getting swollen, filled with. So, here are cells of Rheo leaf. Now, this is also another classic experiment that you will be doing inside your lab probably. So, cells of a Rheo leaf, okay, that means plant cell in a normal flaccid way. Now, if it is put for a long time in a hypertonic solution, this is what we observe. So what do you observe here? The cell walls are more or less intact. These are the cell walls. The boundaries are more or less intact. That means a cell wall. But the plasma membrane has, and the protoplasm, that is protoplasm is cytoplasm with the nucleus, okay? So the whole protoplasm, it shrinks and peels away from the cell wall, right? So this is the way plasmolysed cells look under the microscope in real. Now, one thing we need to remember is that this happens only in living cells. So, in any way, if the cell is dead by wear and tear, or if we boil the leaf, or in any way, if the cells die, this plasmolysis will not happen there. So only living cells exhibit plasmolysis. Now, what are the applications of osmosis in nature? How do organisms use osmosis in their daily life? So unicellular freshwater organisms and most plant cells absorb water by osmosis. Now, obviously, water organisms and plants, they both need to absorb water. And because their roots or some parts of theirs is always immersed in some water or the other or moisture. So that's why they need to absorb that moisture around them. So they do this by osmosis. That means absorption of water by plant roots is an example of osmosis. Now what are the uses of plasma membrane? This one, application of osmosis we looked into. Now let's come to uses of plasma membrane. As we already saw, it helps in the transport of gas, water and fat-soluble vitamins through passive transport, which is also a passive transport. That is, I'll tell what it is. Now, plasma membrane also helps in the transport of some nutrients through processes which take up energy. Okay, so anything that requires the expenditure of energy by the cell, any such process is called active. That means a cell is actively doing it, right? So how is it doing it? By spending its energy. So there are two types of transports that plasma membrane does. One is passive transport, where it happens spontaneously, like diffusion. We saw there are two types. So that is passive transport. Then it also helps in the transport of some nutrients through processes which take up energy. That is active transport. Now another use of plasma membrane in some organisms is that it helps in feeding, in taking up food. So that process is called endocytosis. It's the process by which we already saw that plasma membrane or cell membrane is flexible. It's delicate and it's flexible. So some organisms use this flexible cell membrane to engulf in food and other materials, food and other materials from its external environment. And this is called as endocytosis. Now the classic example for endocytosis is that of the amoeba. It acquires its food through endocytosis. So there's an interesting video that I want to show you. Yes, this video is about how amoeba eats to paramacia. Credits to Mr. Vijay T on YouTube. See how the plasma membrane is able to help it flexibly to taking, like hugging, it's engulfing the food inside, engulfing the paramacia inside, and it's getting digested inside the amoeba. So those are the applications or the uses of plasma membrane transports. Then endocytosis. Now we move on to cell wall. As we already learned, cell wall is basically present only in plant, fungi and bacterial cells and not in animal cells. Right? Remember where it is found? It's found outside the plasma membrane. Okay? So it's the rigid outer covering. It's rigid and strong. We saw that already in Rheo leaf, right? The cell wall remains more or less intact. So that means it's rigid and it's strong. When it's the rigid outer covering that lies outside the plasma membrane in plants, most bacteria, algae and fungi. What is it made of? It's made of cellulose. Cellulose has a complex structure and it provides structural strength to plants. Remember what plasma membrane was made of? It was mainly made of lipids and proteins. Here it's made of cellulose. Cell wall is made of cellulose. And cell wall helps cells of plant, fungi and bacteria to withstand very dilute, that is very hypotonic external media without bursting. Right? As we already mentioned, plants, fungi, bacteria are quite often or mostly exposed to water. And when this water is highly hypotonic, that is highly dilute, there is a chance of the cells absorbing a lot of water and the cells breaking. But cell wall prevents this breakage. How? Because it's rigid, and when the plasma membrane and when the cytoplasm pressurizes the water pressure inside it increases and pressurizes it, the cell wall exerts the equal pressure backwards, inside towards the plasma membrane. So it keeps the cell from bursting. Now the cell bursting can happen in animal cells if it goes unchecked, And why does that happen? Because in animal cells we already learned that there is no cell wall. So in the absence of cell wall, if an animal cell is put into a hypotonic solution for a long time, it leads to cell breakage, bursting of cell, and that process is called as cytolysis. Whereas in a plant cell, it does not happen because of the strength that it gives, it receives from the cell wall. In isotonic, the animal cell remains the same, and in hypertonic also, the cell gets even more damaged, and the whole structure gets damaged. But then in plasmolysis, hypertonic solution, when plasmolysis occurs, the cell wall still holds the structure more or less. So that was the significance of cell wall, right? Now let's review this whole lesson quickly. First, we saw the cheek cell experiment, and what did we observe? That cheek cells have no cell wall, they have nuclei, they are irregularly shaped and loosely packed. Okay. Note that all the points are not about all the animal cells. Then we did a basic comparison between plant and animal cells. Then we learned in depth about the plasma membrane. We learned that it's flexible, it's made of lipids and proteins, that it separates the cytoplasm from the external environment, and that it lies between the cytoplasm and the cell wall in plants. Then we learned how the plasma membrane transports substances, helps in the transport of substances to and from the cell and acts as a selectively permeable membrane. We learnt about endosmosis, which might lead to cytolysis, that is cell bursting in animal cells, and in hypotonic solution. And we learnt how exosmosis occurs in hypertonic solution and leads to plasmolysis. And then we learnt about endocytosis. Now this is the process by which amoeba engulfs the food and how plasma membrane helps it. Then we learnt about cell wall, that it's made of cellulose, it's strong and rigid, and its significance in how it prevents cell bursting and the spoiling of the structure of the plants. So that's it for today. Please subscribe to my channel, like this video if you like my work, share it around. Thank you. Thanks a lot. See you.